Thank you for joining the Once Changing the World, India's first Future Tech Meets Sustainability podcast. And today, I'm delighted and honored to have with me Dr. Rakesh Mishra, who's the Director of Tata Institute for Genetics and Society. He's the Distinguished Emeritus Professor at the Academy of Scientific and Innovative Research and Chief Scientist and Group Leader at the Centre for Cellular and Molecular Biology. His research interests are comparative genomics of non-coding DNA in the context of evolution of complexity, genetic basis of anterior-posterior body axis formation in animals, and role of epigenetic regulation in development. Under his leadership, the Atal Incubation Centre was established at the CCMB, which provides life science entrepreneurs the infrastructure and mentorship needed to encourage innovation. The topic of discussion today is going to be advancing genomics and epigenetics from evolutionary insights to therapeutic applications. So, Doctor, really appreciate you taking time being part of the podcast.
Thank you so much. Pleasure is mine. Thank you.
Thank you, Doctor. So, the human, there's two main things that make us up, a mind and a body. A brain, it's got some 84 billion neurons, 100 trillion synapses, and the way they fire and wire, it brings out a sight, sound, touch, smell, taste. And then there's a DNA, the source code of life, 4 billion base pairs of DNA, all orchestrating, humans into existence. And the Human Genome Project, which started in around 2001, we finally now managed to sequence the entire genome sequence. I believe back in around 2022. We know quite a lot about the human brain. And now we know, pretty much everything about the DNA, or DNA. What, according to you, has been the key learnings in terms of the evolutionary insights, as well as, possibly using that genomic human genome sequence into turning it into data that is useful for healthcare applications?
Great. So that's a very big question. But you're right. Now we have the genome sequence available with it. This has been really a major achievement and it's changing the way we do biology with the science today. And more importantly, or equally important, is the ability to edit the genome. So actually now we know the book of life, we can also edit the book of life and make the book and story as we want in principle. But we are very far from editing the book in a meaningful way, because for that you first know the book. You should be able to read and understand the book. Today we can read the book very easily. We don't understand what is written in this book. So what we know about human genome is this is some over 3 billion nucleotides, and we get one set from one parent, another set from other parent, and all our cell in our body, all the 50 trillion cells that we have in our body have identical DNA sequence. If they have identical DNA sequence, then why some of in cells DNA becomes a muscle, in some cells DNA becomes a liver, in some DNA becomes neurons or brain and things, and in some cells it is intestinal cells that digest food so that we grow and survive. So that's the amazing thing, that a single DNA information set makes over 220 different kinds of cells. And that is the challenge. If we understand that well, then we'll be able to know how this information is interpreted by the nature. And then we can see how things develop. And then we can also see, we can fix when things go wrong. For example, in disease or during the aging process. Why suddenly why we are so programmed to age after, let's say, 70, 80, or why some animals age in 3 or 4 years. I'm talking about only mammals. The mouse, 3 years, human, 70, 80 years, let's say monkeys, 10, 12 years, dog, 14, 15 years. So they are very programmed. You will see some variation, but generally there is a timeline, and this is all because it is species specific, so that means it's written in our DNA. What is also interesting is that a lot more than 100 years back, our life expectancy was around 40 years. Now it is double. It's not that DNA has changed. So that means there's a lot of talk of the environment. And if we adapt well to the environment, with existing wealth of our genome, we can do much better, live longer, age healthier, and avoid diseases and be more healthy if we understand more. So that is very essential thing about it. Of course, coming back to your initial question, I want to just put it that although we are able to read the sequence, we can, as you said in the beginning, human genome, first set was available in beginning of this century, but now, and that took many billions of dollars and many years. Now it can be done in a day with $100, $200. So it's very easy. Now we are sequencing population, not individual. And you will see hundreds of thousands of genomes being sequenced. And that is telling us many things. That is telling us why being all of us being Homo sapiens, why we are different from each other, why we look different, respond to same pathogen. I fall sick, you don't fall sick. So what is the difference in the how our immune system is different? And by correlating these little differences to the respective genomic sequences, which may be causally linked, we explore or uncover what is the information in the genome. Otherwise, just 4 letters, and that's
Right.
all. Yeah.
How amazing, doctor, because I envy you and I envy your job, because you are like a detective of sorts who's kind of peering into a human and understanding, who we are, what we are. And like you said, at the beginning of the century, 2001, the Human Genome Project started and took around 22 years to kind of understand the entire human genome sequence. It started with a couple of $1 billion, and now it's come down to maybe $100. And you said, doing human genome, the entire population. And that's the power of or technology stack, and scientists and researchers constantly kind of asking questions and pushing the boundary of the frontier science. I want to know, yes, we can read genome. How do we how do we get to understand this, because I think once we understand 3 billion plus base pairs of DNA, I'm sure there's a whole lot of things that you can do. And you also spoke to the identical DNA. Every DNA is pretty much similar, 99.9% across the human race, and programmed. You use the term programmed, that how it's programmed at a certain point of a life linearly. We start aging, and because of the age, there are these cascading effects of diseases starts coming. And then you also pointed out to the environment, the epigenomes, because there's this, not just the DNA, the epigenomes also play another role. So I would want to understand from you that yes, we can read the DNA. What will it take for us to understand the DNA? And basically, what is that thing which is still needed for us to understand the DNA?
Yeah. So that is an important question. And people have been doing this in different ways for many, many years, for centuries. And one of the branch of biology which we are talking around is, we call it a developmental biology, which means how from an egg, whole organism develops, how from a seed, whole tree develops, and then the rest of the associated characters that it acquires. So, for example, and then we cannot do experiment with the humans because that's unethical. And besides, we will need very large number to figure out anything, because it is like hitting in the dark, and suddenly when you get the aim, then you go and look what was there and so on. So you require very large number of organism if you want to do something. So that is why geneticists have, biologists have adopted approach called model systems. So they use some organisms which are easy to manage. For example, we use in the lab Drosophila melanogaster, a fly, a fruit fly. All of you have seen. If you leave your banana or some fruit in the kitchen or a dining table, you will see a little creature flying around, very innocent, harmless. But that's the fly that we use in the lab for our experiment. And people have been using this fly. In fact, it has given many Nobel Prizes. Lots of basic discoveries have happened because this is a very convenient model system. Life cycle is just 10 days. and it also has a head, thorax, middle of the body, and the abdomen. And what people figured out using fly as a model system, that the basic principles of development, that how eye is formed, how anterior posterior body axis is decided, that where will be head, where will be tail. This basic principle is sort of common in all animals. Like same set of genes make head in fly and head in human. It's just that fly head will be different from human head, but which end will be head and which end will be neck and where tail, and where will be the neck, and where appendages, where our hand will start to grow, where our legs will start to grow. All this is written in our gene, and principle of those are very, very common. Developmental mechanisms are so well conserved from in fly to human. So if we study flies, we start to interpret the human system as well. So, for example, if we say that, okay, this gene is important for deciding where the forearm should grow, is that gene also there in humans? You go look, yeah, indeed, it is there. And then are there then some other animals where this gene is? Then we see that it's everywhere is found. And then you start seeing one of the approach geneticists use is inactivate the gene, mutate it, and then ask what happens. If you have taken the function off, like if you take a car and take out the wheel, then it will not move. So whether you know anything or not, but you can tell that wheel is some component in this object. If you remove it, it will not move. So now you know that movement. Then it will take some time to say a wheel is connected to axle, wheel is round in shape, it is filled with air, so it helps. All those details you will figure out. But knocking out a wheel will immediately tell you the functional importance of the wheel. Similarly, if you knock out a gene, we will know this structure is not formed. If it is not formed, then you go start. Now you have got a breakthrough. So now you start detailing where is the gene expressed, what are other proteins that interact with this, if there is any environmental impact on the in different individual, this is different, and so on. So you go into many different kinds of details of so why we are so crazy about genome population? That is something because now if you sequence lakhs and lakhs of individuals, now you see that there in some population where some people have more cases of diabetes, some places there is more deafness, some places there is in some communities there is a more certain genetic disorders, and this is very common observation, and also big problem for a country like India, because we have over 55,000 distinct ethnic groups, which means these guys, they follow broadly, they follow endogamy. There are consanguineous marriages, and so that means if there is a mutation that is being enriched in the same population, and now there are several thousand diseases which are being now associated, which are community specific. So that means nature already has now thousands of mutations available. You can go and take some cells from this individual. Now we can grow the cells in the lab, make induce pluripotent cells and make organs and study lots of things. So instead of going the way we do mutagenesis in mice and flies, the genomics has empowered us to study similarly thousands and thousands of mutations in the human population. That's why genomics has become so important. Everything doesn't have to cause disease. Sometimes some metabolic pathways are faster in you and slower in me. So that means if you take the same drug, you will metabolize it very fast. So you need maybe 100 milligram to have some effect. And by afternoon, you have all processed it. My metabolism for that is slow, because that enzyme has some mutation, is functional, but it is slower. So that same amount of drug will last for 2 days in me. So that means I need really half of that dose, okay, otherwise I will feel the lot of toxicity and discomfort. So this is called pharmacogenomics. So studying various mutations, or what is a single nucleotide polymorphism, that is how we are different from each other, and associate what kind of genetic signature means that you should get more of the same drug or less of the same drug. You will be surprised, or maybe you are aware, that half of the chemotherapy drugs and more than half of neurological disorder drugs don't work in the people. So if you give to someone chances that it will work for that person is 50%. Why? Because that person's genetic signature is different. If the target are mutated, so the drug is not working there. So now what is happening is we are collecting genomic sequence of individuals and associating that this drug will work for you and this drug will not work for you. So when next patient comes, we take first the genome, look for the signature, and then decide which drug to give. So that means not only we save in half the people the torture of given a drug and not having any outcome of it, and save 50% of the money, and then give them the right drug which they require. So this is called personalized and precision medicine, completely dependent on genomic signature and associating it with the response to various kinds of drugs. We can also even predict to some degree what is the possibility that you will get this disease or that disease, and how by moderating your lifestyle you can have a better and more comfortable and healthy life for a longer period of time. So genomics is really going to be like your ID card. This time I'm not really speculating beyond reality. In few years, we all will be going to hospital, and first thing they will ask is, where is your ID? So the genomic card. And then based on that, they will prescribe drug. You will get half the dose of paracetamol, I'll get double the dose or whatever. Even simple thing. People will laugh at us after 10 years that how you're treating without knowing how much to give and how much not to give. So the genome is going to be really, by the way, without even understanding much. It is just data now. Because you sequence 1 lakh people, 50,000 people did not respond. Look for what is common in their genome without knowing whatever it is. It's like saying something that is green, maybe it is a plant, and something red, this is something else, and so on. So just without understanding, but the data's power is so much, and the machine learning is coming to play, that more and more data is collected, more and more of phenotype or drug response is recorded, and we just mix and match and find out signatures, do some more analysis of the data set, and then you start giving. We will understand about how it works later. If we understand, then we will be able to change those things and make it better. Today, we are not educated enough to read and understand the book of genome and make any. For that, we are largely illiterate. We can read it, but we don't understand what is written in there.
Right.
So that's an exciting thing ahead.
Yeah, super. I'm so excited. You mentioned that, we humans, we are same, yet so different. And it's the environment that, which plays a role in individual, genetic signature. You spoke about personalized medicine earlier in the course of the conversation. You also mentioned that, we've kind of figured out how to read, not just read. We figured out also to write, to delete and add, a specific genome, that's CRISPR-Cas9. And there was like another genetic, revolution of sorts, Will you be able to kind of elaborate on this CRISPR-Cas9 gene editing? And you spoke about personalized medicine. It will be great if you could, share the role about CRISPR-Cas9 in drug discovery, age reversal, and other targeted therapies.
So CRISPR or genome editing technology in general is something extraordinary. It's just simply amazing. There is no adjective for me to describe the kind of power it gives you. Imagine we evolve over millions and millions of years. A mutation is naturally occurring, and then environmental selection pressures operate, and then it gets established in the population. And it takes so long time. Today you can go and make in 1 generation, and if you understand what to do in 1 generation. So you are like, if there is a concept of God, you are going to make whatever you want to make, right? If something of that sort was making organisms around. So it's that kind of imaginary thing you can actually do today. So for example, you can, one of the field where genome editing is going to come to public use faster is genome editing in the plants, because that is in many countries allowed. Even in India, it is allowed to do genome editing, and that is not considered GM, because we are not introducing any foreign DNA in that. It's just 1 or 2 nucleotide changes. So by doing this, many labs, including TIGS, are making crops which will require less water, which will be resistant to disease, which will be resistant to pest. So now you don't need various fungicide, insecticide to spread. You don't need so much of water, because water is fresh water that is required for, let's say, for growing rice is very large, and it's going to be a problem in future. So by just doing genome editing, you go do a precise change, and then you have what you want. So people are even imagining of something called designer's crop. So you will have a crop which will have more protein, more zinc, more iron, low GI index, so that it is good for diabetic people, and it will have good yield. It will be resistant to pest, resistant to disease. It will require less fertilizer. Therefore, soil health will be protected, less water. Because all these properties exist in different plants. There are plants that go in desert. That means they can manage in very little water. Go in your garden, you'll see some plant is eaten up by the insect while other is thriving. So why insect did not like the other one? Because the other guy has a protection mechanism. Now we transfer that here and so on. So more we understand things, then we can start making the crop. So because if the world population needs to be fed, particularly nutritious food, we need to make lots and lots of changes in the way we are doing agriculture now. We're not costing environmental costs. We are not costing soil health deterioration, those kinds of things. So we have to address that. And particularly, if we know things better, then we can manage to handle the, the crisis which will come because of the climate change and global warming. So these are the strength that this technology in just past few years has given. In for the humans also, we are thinking there is something called gene therapy. So that's available for some diseases. They are generally rare genetic diseases, and in which it's very prolonged process and very expensive. In Indian rupees, it costs some 20 crore or more for a disease. So you can imagine that even by any means, I don't think most people can afford this kind of treatment in India. So, but then there are easier ways of doing that. Now there's something called in vivo gene therapy, in which you just now said. So now you take CRISPR-Cas9 mRNA, which will make protein later. So take this mRNA, make a guide RNA, which will target where CRISPR. So guide is RNA that will guide the CRISPR protein to go to a particular location in the genome. It will go and cut the DNA there. Then you also provide a template. So you're now one lipid nanoparticle, little bit like SARS-CoV-2 vaccine of Moderna and all the same technology. Now inside you don't have mRNA of virus, but you have SARS-CoV, you have a CRISPR-Cas9 protein that does editing, guide RNA that targets protein where to go, and template which will help in repairing. So you go and actually repair the defect that was there. Once you've repaired, all these are not genetic material. They will make this RNA with some protein. After that, they will go away. But now DNA is repaired. So when that cell divides, it will have the correct gene or good gene. So you have fixed the problem just by one injection. So imagine if you inject in the muscle and you have muscle disease, and 20-30% of the cells start to make the protein because they are corrected. For lifelong, now you don't need any drug or anything. Imagine, so that is the future. That is the future of medicine. That is the future of personalized medicine, because we have to go and read individual DNA in individual person, what is the defect, and then design the thing. And the way technology is progressing, these are the things that are not going to cost crores, not even lakhs. So they will be really affordable, out-of-pocket approach, because that's what most of us in India do our medical care with that way. But technology is very, very promising. The Moderna vaccine production cost is less than $4. So that means we can do such treatment in maybe, let's say $10. Produced in India, it will be less than $1, because we have production cost much less than it. So it's very, very interesting how technology is changing the course of healthcare we know more, we can analyze better, and we can do more precise interventions because we understand those things.
Right.
It is a process. I'm not saying that we have arrived everywhere, but we clearly see the path.
In the course of the conversation, you were mentioning about your works with fruit fly. With fruit fly, there is, besides the DNA learnings, there is also a group of team that's working on the connectome, where they are trying to understand the entire cognitive structure of the fruit fly. Will be great if you could kind of explain, Hox complex and your works around fruit fly, and if you can elaborate how these genes orchestrate the formation of different organs and body parts.
Hox gene that you mentioned, they are also homeobox containing gene or homeotic genes. These are a set of some 10, 12 genes that lay the plan of body axis in embryos. Early on in the embryos, they decide where will be the head and where will be tail, as I was talking in the beginning.
Yeah.
We know that because if we mutate these genes, take their function out, and now particularly with CRISPR, we can go and mutate whatever we want, whenever we want. So we can, if we mutate a gene which was making, let's say, legs, now there will be no leg. Or we go and ectopically express a gene that makes eye, expresses in the leg, then you will say that eye is found in the leg. So we know that what is the initial gene that is triggering the function. So similarly, for the body axis, there are something very peculiar that have actually, for this, Edward Lewis was given a Nobel Prize, and there were two other developmental biologists, Nusin Falhard and Eric Bischoff in Princeton. Nusin was in US in Tübingen. And so these three people, they did a lot of genetic. What Ed Lewis' work was that there are genes which are on the chromosome together, one after other, and there is a collinearity. A gene that one end makes a head, next one will make neck, the last one will make tail. So that means a chromosomal organization of the genes, which we now understand is homeotic gene. Not only they are arranged in certain order, arranged in cluster, they are also arranged in certain orders. That gene 1, 2, 3, 4, 5. 1 will make head, 5 will make tail. What is amazing is that the same thing happens in mouse, same thing happen in human. Gene 1 will make head in mouse or human, and gene 5 will make tail in mouse and human and fly. So these genes have their names. So just to keep it simple, so which means that the fundamental strategy, how in whole animal kingdom head and tail is defined, there's only one way. Nature knows only one way of making head and tail. If you understand well in mouse, we will know understand this in to large degree in other organisms. So if we know this, then we can make, so for example, I want to convert a fly, which is generally 2 wings, I want to make it 4 wings. So I go and I know what makes the wings, and I go and make that change. And now you will see that fly has 4 wings. I can make longer fly, I can make shorter fly, because body segments are, we know which gene is making which body segment. Now we can go and replicate, duplicate those or reduce those. So I'm just telling you the basic thing that now if we go and change this gene region or this cluster called Hox cluster, scramble the genes, their order, we can predict what will be the outcome for the embryos. So this is how the large number of people have worked in. Our own work was to understand how a gene stops to expressing in one place and doesn't go beyond that. What are the boundaries that decide? And once the expression pattern is set, what are the memory elements and how they function? That after in the initial proteins that set up this expression pattern, they go away. But the expression pattern of whatever master control genes they have set up continues lifelong. So each time cell divides, they will reestablish the same environment of expression. So what is that memory element? And if we understand better, then we can understand more about aging process, because as cells age, this memory is what loosens, that is general understanding. Because after 1, 2, 4, 10, 100 times, that memory is not there, this molecular environment changes and it becomes loose and lots of genes get misexpressed and we age generally. That's a very broad way of saying. If we can tighten that, we can do some kind of servicing of the locus in a way that it is now like we do with the cars. It's also fresh again and engine is fully tuned. Then we can prolong the healthy cellular states and therefore reduce the cellular senescence and therefore the aging process. Aging, because it's a development from one egg to adult is one, but then all the function and keeping it functional to some time, and with respect of time, switching it to now it is no more required and aging, it becomes a very interesting part of the biology. It's also part of developmental biology and it's very relevant because in countries as they develop, although India is entering into that phase, lots of people with 60 plus, 70 plus, and they are age-related diseases and disorders. So that means that disease load or healthcare load is increasing like anything in many countries, developed countries particularly, because people are living for a very long time. So all kinds of health issues come, which is not there in younger people. So how to reduce that burden in the healthcare system? If we understand aging better, we can make those people healthy. They don't have to depend on medical devices or hospitals and so on. So that's why, and this is essentially basic biology is the same. So there's a lot of interest now in the understanding the aging for many reasons. I mean, people want to live longer, but more important is how to age in healthy way and fix certain issues which can easily be done once we understand the process.
Right. It's so very interesting. Yes, you rightfully pointed out. I think aging is such a problem because, I mean, though it's natural, but after a certain age accumulates diseases, and there's a certain aging population which can, I mean, with this technology stack, I mean, the accumulation of senescent cells is one of the reasons why we age. And if we kind of understand that and reverse that, I think it'll be really cool. You also mentioned about your works with the Hox complex, and I mean, with changing maybe like understanding the dynamics of the DNA, maybe changing the head to the tail. There are various researchers, scientists around the world, and one is Michael Levin, Tufts Laboratory. I think he's pioneering something called as bioelectricity, and he's working with these planarian worms. And he's taking the head and putting it in next to the hand. He's creating planarian worms with multiple heads, multiple hands. I think this understanding of biology will help us not just augment ourselves, but also find ways that you mentioned about possibly age reversal and other opportunities. These understanding and research in Hox cells, Hox complex cells, what are the other benefits in the healthcare that you see happening?
Oh, I mean, it's so many things now, the new technology and understanding, as you mentioned, biological systems do things and we get inspiration from them. Like biological systems can produce food, they can produce heat, they can produce electricity, even magnetism, all kinds of things. So for instance, we see all around us greenery, right? So how it makes? It is because it captures the energy from the sun. Rest is all recycle, but only energy that is required is taken from sun. Then we do very inefficient way of converting this energy that has been captured from the sun into various kinds of things, the coal, petrol. We don't convert, but the nature has done. And now we burn that and ruin our environment. Why we can't work like plants? Straight away use the solar power and do whatever you want to. And I'm sure in next 20 years and so, we will be using 70, 80, maybe 90% solar power for all what we do. So, and this is, so we are late learners. Plants have learned it very early on and established this beautiful planet called Earth. Because if there's no photosynthesis, nothing will be there for anything to eat. No organic world will be around. So one single mechanism of plants has made life possible. And we have completely missed it all along, or deliberately, or for whatever reason. I don't want to go into that direction. We have avoided that route and still used a billion year of protocol to convert a plant into petroleum and then use that petroleum through our motors and then use it for our whatever energy needs, whether you burn to heat water, or you put in petrol, or put in your car, or all kinds of things. So if we didn't go that billion-year routine of making petrol, we straight away use sun, learn from the plants how they are doing. We have a technology in our hand, we can do, and now it is becoming more and more. So all of us will get fresh, fresh. I wouldn't say, but free energy, like plant. So we have not learned from biology. That's how we have disturbed this ecosystem. If we function in a biological way, we will be more tenable and sustainable. Otherwise, we will not be. If we do the way we are doing, we are going to destroy ourselves very soon. Right now, these are not this happening. Now coming to so that is one maybe very dramatic way of pointing out what all we can learn from biology. But now the technology is coming in such a way for the health. For example, one thing which is becoming very important is prevention is better than cure. We are hearing this wisdom for maybe centuries, but we don't practice. We when we fall sick, we go to hospital and get those kind of things. But if we have good vaccines, then we don't have to go for any infection. We can prevent that. So now you have seen the revolution in vaccine biology, vaccine technology. mRNA vaccines have really transformed that. They are safe, they are cheap, they are easy to make. You need not now decades, not years, not months. You need weeks to make a vaccine. So that is something, if you ask me 3 years, 4 years back, I would laugh. Somebody, you can make vaccine for a pandemic? No way. It takes 10 years. But we use vaccine for. So that's how the technology transforms. And sometimes it's so good to be proven wrong, because we become traditional after some time. So and the new technology changes our perspective. The other thing is surveillance. Now, by just taking the wastewater that is being running underground in the cities, we can tell you what is the pathogen status in this city. More granular you go from where the water has come, it may even go to one colony, one complex, and we can tell how many people are infected with which disease there. So that's the power of environmental surveillance, which has were there before. Now with the newer and newer technology of DNA processing, RT-PCR, and various other sensors that detect DNA sequences and various other things, we can actually give a very good picture of what is the pathogen status. So most of the disease that we carry in terms of infectious disease, they are asymptomatic, which means the symptoms are not so much that we even notice. We may feel little tired, little here, there, which is we don't know why. Sometimes being angry is a bigger symptom than being infected by something. So our mind dominates many times. So we don't know whether we are infected or not. Is not required also to know all the time. Otherwise, we'll be constantly worried. Our body carries more bacteria than our own cells, several hundred times more cells of it. So in a way, we are a house for bacteria, not independent individuals, as we imagine, as one way of. So, but what now we know is that how the pathogen status is there in the particular city. And when we do longitudinal analysis, now this is becoming really a permanent feature of every municipality and part of one thing, what is called One Health. So this has to be constantly monitored, and we will know well in advance using mathematical modeling which disease is going to have a outbreak, when it will break out, and when the number will increase. We might be even able to perceive well in advance the potential pandemic candidates in the future. Lots of things are coming like. One big problem is antimicrobial resistance. Using wastewater, we can tell what kind of resistance is there, whether it's coming from the misuse or use of antibiotics in poultry, in animal farms, or in fisheries. There are lots of antibiotics are used in this, or use or misuse of antibiotics in human. So from where this resistance is coming, and so we can go and address the root cause and not the symptom that income. So this kind of very sensitive way of and by doing zoonotic surveillance, which means surveying from the vectors which interact with the animals in the forest, let's say, like flies or ticks, and or isolate pathogens, the animals that are interacting with the forest area, and then we know what kind of pathogens are there, and if something is coming closer to human, like you may be hearing these days H5N1, which is the name of the virus for the bird flu. Now, the bird flu, birds can spread here, there, only birds are bothered with that. But now we are seeing that this virus is infecting some mammals also, some cattle also. There are some 1 or 2 human cases also. But only thing is the human-to-human infection is not there. But if human-to-human infection of this virus happens, we are in trouble, because then the virus is infectious, but we are not able to transmit from one individual to another, only if you have to go through bird route, so we can avoid at the moment. But if viruses evolve, like we have seen with SARS-CoV-2, how every 2-3 weeks we new variant. So these are the potential threats. So without any symptom, without anything, by just monitoring them in very precise level by genomic surveillance, we can see how it is changing, and we can see whether the threat is coming closer to us. And more importantly, since we understand the root of these viruses at the moment, we can cut the pipeline of the virus entry into the population and preempt things from happening. So we can know, without knowing, we are walking in the dark. By knowing by this surveillance, now we know what to avoid and where is the safer way. It's like walking on the road. If you have, if you don't know, if you don't have visibility, you are going to hit something sooner or later. But if you have visibility through surveillance, you can avoid things and have a more healthy and avoid economic disaster that happen when disease is out.
Future looks so exciting, Doctor. Really thankful for you to share all these insights. I'm not going to take much time of yours. Just maybe 2 questions. One is in the space of cloning, because there is pioneers such as Dr. Church, who's working on something called as the De-extinction effort. Where they talking about like bringing back the woolly mammoth, the Tasmanian tiger, and they're also working on cloning primates. Using somatic cell nuclear transfer for potential human organ donation. So what's been the scientific progress in the field of cloning, and are there any Indian scientists working on the space?
So cloning is very now kind of, if I can say, not anymore surprising or something very difficult. It is now a regular, different kinds of cloning. It's sort of becoming routine. So I mentioned pluripotent cells. So iPSCs are induced pluripotent cell. You give me blood from a human or any living cell. In 2 months, I will convert into iPSCs. So potentially, I have now material to create that human again. Whether we should do it or not is a big issue, and I'm not in favor of cloning humans, because life has its own beauty, and without understanding fully, we start making. It's like you don't know the engineering of the car and you start making a car. So it will look like a car, but it will not run because inside you don't know what is the engine. So that is how we have understood not so much of biology that we can go and create a new species. But certain things like in agriculture, cloning is very common. In fact, lots of agriculture in the tissue culture lab, they are cloned. And then the farmers are given those little saplings and they can go and use. So you know exactly the variety because they are all clones. There is no variation in them. So this is happening in some case. It will happen maybe in some food items and all, because once we have established something, you can, in fact, grafting of the plants of the mangoes, and that's also cloning. You're taking from here and the same plant you are growing in many different ways. So cloning essentially means same individual, identical genetic structure is being used for, and we are preventing the sexual reproduction, which is the essence of evolutionary mechanism to drive evolution. So if we block the evolution, we are blocking our future. So we shouldn't go beyond a point where we use an end. We should not clone something which is going to go in future over which we have no control. We cannot live in future. That we have to leave it for next generations. They will decide when they will know more about it. But so the process of cloning is a strong, like for cell therapy, we need to clone cells, take a cell from patient, for example, which has disease. Now fix it using CRISPR technology and genome editing and make the cell now healthy. And clone these cells in the sense, make large number of those cells and put in the body, and the person becomes healthy. In fact, this is essentially partly what is a gene therapy. So that's being used. But cloning individual and primate and de-extinction, I have serious concerns about. Extinction is not because somebody went and killed the animal. Extinction is because the environment changed. We have to, if we are serious about conservation, we should conserve environment. Biology is powerful. They will live on their own. You can make a mammoth, but where will mammoth live? You can create all the old, for example, somebody may come with the great idea of making dinosaurs. Imagine if you can do what will dinosaurs do? Where will they live? We have ruined the forest. We have converted them into field and houses and whatever. If you don't have a place for them to live, if you don't have food for them, there is no prey, and prey will need food. So we need that for conservation, not making that is just fancying. So conservation, I have very different view. You can make that animal and put in the lab or put in a zoo. I don't know whether it's ethically correct to bring some organism just because organism cannot question you and put it in a jail for your fun, scientifically, maybe to understand more. So that's something one can debate. But for research purpose and understanding purpose, within ethical constraints and permissions, this should be done. But we should not venture into environmental intervention, because we simply do not know how many species are there on this planet. Very few of them we have. And we are going to change that without knowing. Just because we know my office, I'm not going to change the whole Bangalore. I haven't gone and understood about my office, I can change. So if anything goes beyond my office and building, I have no authority or moral rights to change. So that should be very, very, I'm sure better sense will prevail. But since you ask questions, I think that should be done with lots of care and control.
Doctor, my last question to you. There are a lot of students who would want to kind of, get into the field of biotech. What would be your advice to them? And what's that one moonshot that you are going after?
So first, for the new students, I think this is the century of biology. Today, also, I think most countries, particularly developed countries, healthcare economy is 30% of the economy. So that means, and is going to increase. As I was saying now, agriculture has to adapt to climate change and environmental factors that are changing. Healthcare, as we discussed, some aspects is changing. Infectious disease are going more north because of increase in temperatures. Vectors are going more there, so the vector control. So all these things are posing huge problems, and therefore, and the technology of we can catch any organism, take little piece of tissue and understand the complete genome, and we can change the genome we want. So we have all the tools. We have nail and hammer. Now we have to find correct place to go and put the nail so that it is useful, right? So it's that kind of situation. We are technologically enabled, and the data that is generated, that is also amazing thing. For many young people, you don't have to do anything. Just go and analyze the data. You can come up with now these AI and machine learning. These are so powerful that by taking the data and whatever metadata is related to that, you can come up with very interesting hypotheses, observations, and ideas that can be then tested in the laboratory conditions. I would think that by understanding the genome sequences functionally and able to correlate different genomic signatures with our disease response or conditions, we will be able to manage health to the best of its biological potential. I'm not going to change anything, but I will understand. So this can be done so that people age slower, rare genetic disorders will not be a problem because we know who are the carrier, and we can tell before the birth very early, within less than 3 weeks or 4 weeks, what the baby is going to be. And if it is untreatable disaster, then disease, and the child is going to be disabled for next several years, for 5 years, 10 years, 12 years. There are various diseases in India. More than 90 million people suffer from one or other of this kind of diseases. They all can be, if we do the proper carrier analysis, the birth of such babies with such problem can be prevented. So imagine we are adding 1 million every year such disease children. We can prevent that. So essentially, by understanding and using appropriately genome information and genome technology, we can have better life, which is disease-free largely, or better managed disease, and healthier food to eat, and a safer environment. Without destroying our environment, we can grow food. Right now, we are growing food, but we are poisoning the soil by so much of insecticide, fertilizer. All that will soon be preventable. So, right. Yeah, long, long list, but and all this we require workforce people to do. And it's not only development in life. All these technologies then need to be taken to people. And so lots of biotech industry is required. And India is government has now lots of emphasis on biomanufacturing, because they are going to be the future. They're going to replace a lot of current methods of treatment and agricultural practices. So young people in biology have great future. Is that they should not always think, I will do PhD, I will be professor, I will do. Take innovative path. The moment you have technology, you are smart enough, study well, connect with some mentors, incubation centers, and take a route and become rich.
Doctor, really appreciate you taking time and being part of the podcast. The conversation was so insightful. I'm sure the listeners are going to enjoy. Yes, we're sitting in a fantastic point of time where we've got to learn from the nature and be humble rather than be arrogant. I think that's, if we understand that, there are so many things which we've been doing wrong, we'll be able to correct, and we'll get into the age of abundance where we'll be able to, one step at a time, maybe, start reversing, or health issues or aging problems and stuff like that. And in that effort, we will also make the world heal, not just us, but also the world, which desperately needs at this point in time, because of a consumption without conservation. So really appreciate taking time and being part of the podcast. And to my listeners, if you like what you see in here, then please press the subscribe button. And until next time, see you there. Thank you, Doctor. Really appreciate this.
Thank you. Thank you very much. Thank you, Eddie. It was very nice talking to you.